Hi, welcome to our first STEMinate podcast. My name is Mrunal Kute, and I am the current STEMinate Executive Manager. For our very first podcast, we will be interviewing STEMinate and Illuminate Journal founder and current Duke student, Perisa Ashar. Hi, Perisa, how are you?
Hi, Mrunal. I'm doing well. Thank you so much for interviewing me on the first episode of the STEMinate podcast.
Of course. So let's jump straight into the interview. So for our very first question, how did you get involved in science fairs?
So thank you for asking this question, Mrunal. As a kid, I've been always fascinated by STEM, and I think I owe a huge amount of inspiration to our local Math Science Innovation Center based in Richmond, which offers STEM enrichment opportunities to students in elementary and middle school. And from there, I remember I attended a camp called DNA Detectives and how to basically extract our own DNA. So that basically sparkled the idea for my very first science fair project, which was the effect of different cell lysate temperatures on the amount of DNA extracted from strawberries. And I remember completing this science fair project back in seventh grade. We had basically transformed our kitchen into a laboratory. And I remember like it was so fun, this whole process of scientific inquiry. And after attending the local MRSF or Metro Richmond STEM Fair hosted by the Math Science Innovation Center and learning from other middle school projects as well as high school projects, the following year, I basically had a hybrid project both conducted at my home and my first experience in a laboratory where I basically tested the effect of different mushrooms as novel inhibitors of an enzyme in pineapples called bromelain on their degradation of just skim milk proteins, which I conducted again at my house through ordering petri dishes. But when I did further research and found out that both mushrooms and the bromelain enzyme that I was analyzing at my house were also anti-cancer agents, I remember sending numerous emails to various VCU faculty, and I finally got a response back from one postdoctoral fellow, Dr. Praveen Bhupathi, who allowed me to test these anti-cancer agents, just my homemade solutions, on prostate cancer cell lines, which honestly was so cool. It was the first time I was able to set foot in a laboratory at the age of 14. And honestly, none of my parents are in academia right now. And so kind of having the opportunity, I was just so grateful for, especially, I didn't really know this postdoctoral fellow at the time and he gave me a chance. And after I've conducted laboratory research in eighth grade, I felt more inspired to not only do mentored research in high school, but also other independent research. So in high school, some of my projects have ranged from analyzing hyperpolarized currents in mice thalamus neurons under sedation or mice exhibiting autism spectrum disorders to looking at blood-based biomarkers and tumor biomarkers for cancer, not only through in vitro analyses, but also through a plethora of bioinformatics analyses from the Cancer Genome Atlas database, and then subsequently machine learning from histopathological images.
Wow, that sounds amazing. It was so great hearing about some of your first experiences with research. So kind of as a follow-up, what else have you conducted research on, kind of going into the specifics?
Of course. So after the strawberry experiment, I first looked at in eighth grade the effects of different mushrooms, as I mentioned, lines proteolytic, or in essence, basically that means its ability to degrade proteins, in this case, skim milk proteins, and then testing their anti-cancer effects on prostate cancer cell lines. And then in high school, I was able to do and partake in more mentored research, my 9th and 10th grade project, which looked at the effects of various sedatives, or in this case, just one sedative called dexmedetomidine, on these hyperpolarized currents in mythalamus neurons, brains, which are important in essence for relaying of information. That's what the thalamus is crucial for. And then after that, I went more in the cancer realm and basically was able to learn a lot from interpretation of next generation sequencing analyses, learning how to do various protocols in the lab, ranging from PCR as well as helping out with other experiments like Western blots, um, changing cell line media, and then during the COVID-19 break, when all labs are closed, then doing my own independent analyses of gene expressions in various lung cancer patients, from squamous cell carcinoma to non adenocarcinoma, and looking at the effects of basically different types of patients ranging from different races, sexes, smoker history, you name it, on these microRNA expressions. And then ultimately after that, getting more into machine learning and how we can help do automated detection of cancer through just tumor-based images.
Wow, that sounds very, very interesting. So, what has been one of the most challenging aspects of research so far?
What a great question. Honestly, I've just been so inquisitive. So, like, at times I'll
Mhm.
just be walking, just thinking about, we could do this, we could do this. Oh, this could be a future step. And I would just say, especially when you're younger in high school, it's really hard to get a lot of these opportunities, right?
Definitely.
But in a laboratory, like, you have all of these ideas, and you've after
Hmm.
you've done the independent work, like computational analyses or something at your house. Like, for example, what I did with my bromelain experiment, I did, the first initial analyses in my house, looking at the proteolytic effects, or getting that initial set of data. And then even if you've like worked really hard and you have all these good ideas, being such a young student, it's often hard to get that opportunity to participate in especially laboratory-based work. So I would say one challenging aspect is kind of moving forward with like independent ideas,
Hmm.
like an official lab. And my best piece of advice would be to just get involved with other projects or mentored projects occurring in the lab at that time. And then, if possible, really explain your interests and how you can tie that into what you want to study. Um,
Right.
and then try to do something on the side too. Like, I did also some computational analyses for the lab to show them, how I could help in that aspect, in addition to performing some in vitro experiments at the lab. But I would say this problem really goes away when you're in college, which I just, was kind of relieved, because, like, you get
Hmm.
many opportunities, and it's at that point expected that a lot of college students, depending on what field they're in, like in the life sciences field or medical field or even engineering field, they are usually involved with research from the start, and
Right.
it's not hard then getting opportunities to access a lab. However, I
Hmm.
kind of tell everyone that at this point, like, I'm again so grateful for the experiences I've garnered in a laboratory setting, but actually, especially this past year with the COVID pandemic, you don't really need a lab to often carry out a really good science fair project.
Hmm.
One of the science fair participants, who I read her work on, basically conducted this experiment with fruit flies and looking at the brains just through her house, like laboratory setting, similar to how I did my initial projects in the science fair, and still found a novel finding. And I think at the end of the day, that's what really matters, like what you're able to do with what you're given with, and to the extent of how far you can
Right. Right. So, kind of flipping this question around, what has been one of the most rewarding aspects of research?
go. The knowledge. I think that is the best answer, because when I, just started, again, just reiterating, I had like no like prior experience. I was learning along,
Mhm.
and I
Yeah.
wouldn't say this is just a journey where you just immediately know what you want to go through, like jumping right in.
Hmm.
For me, I'm very grateful to my mom and dad. They've really supported me, and like, dropping me to all these different like laboratories, or helping me download programs. But at the end of the day, independently teaching yourself, not only just the scientific aspect, right? Like, I was able to learn so much about the brain, in such a electrophysiological level, that I thought I would never learn, when I was learning different procedures, like helping with the whole cell patch clamping, or learning how to genotype mice, and analyzing Western blots from there, all the way to then the cancer realm, learning how to help out with other experiments, as well as conducting my own research, participating in laboratory meetings, learning how to, discuss findings and receive feedback, and as well as learning how to write more formal manuscripts for potential peer-reviewed publications. I would say it's not just the science, but the other aspects that come with it. Trying to find a mentor, like reflecting on what you could do next time, learning how to find mistakes, learning some of the differences on how to really make sure everything is good, before peer review too, when it goes to that step. And it's been an amazing journey so far.
Mm-hmm. Yep. Okay, so you are one of the founding members of STEMinate. So what has kind of been like a catalyst, or what has encouraged you to start this organization?
Oh, I remember. So in 10th grade, as I mentioned earlier, the Math Science Innovation Center has been, such an important aspect of, I would say, not just my life, but many other lives as well, not only through the enrichment programs, but through the Metro Richmond STEM Fair. And one thing that really kind of was the catalyst was when the local Henrico County and Chesterfield governments decided to discontinue funding for the Math Science Innovation Center. At that time, me and a few other students and parents were very passionate about continuing it. I remember leading a petition. I spoke at several educational board and board of supervisor meetings, especially for the continuation of the STEM fair. And fortunately, they were able to allow some funding, for the, resuming of the STEM fair, but not back to the original amount, which really cut a lot of the STEM programs offered by the Math Science Innovation Center right now. And ultimately, they were not able to really deliver these programs at the scale they were before. So I created STEMinate to basically follow the same modus operandi of delivering complementary STEM education to students from all communities, including underprivileged communities, either through in-person workshops at first. So it was more like a very local-based initiative, and then I was able to grow it to an international nonprofit by ultimately hosting seminars, summits on a variety of topics relating from experiments conducted aboard the International Space Station through collaboration with other existing organizations like the Virginia 4-H and Henrico 4-H, which we've been very grateful for as they've helped us with a lot of the funding and carrying out our programs to a larger audience. And I feel like at this point, I'm just inspired to take STEMinate on as this big umbrella organization with various sub-initiatives under it. Like we have the Project Infinity initiative, and now we're also going to develop a computer science initiative too, where we're planning to host a hackathon next year or the year after. And I just think I'm just excited for what the future holds for STEM.
Wow, that sounds wonderful. Okay, so kind of switching up course. So you are a BME, or a biomedical engineering major at Duke University. So what is some advice you can offer us or share with us today?
Honestly, I've learned so much being an engineering student while on a pre-medical track at Duke. It's a very interesting major, just obviously it's only BME, but the realm of engineering. Like in high school, as you may have mentioned earlier, I did a lot of computational biology, bioinformatics research, and in vitro experiments too. So you could say I did more of like cellular BME. Like BME is such a broad field. Like it could range from working in wet lab experiments all the way to prototyping. And so one thing that kind of really jumped out to me at Duke was how integrated we were as BME majors with the other engineering majors like MEC-E and ECE, which basically stands for Electrical and Computer Engineering. And in our 101 class, we're basically working with these other engineers on delivering an engineering solution to a client. Like we go through this very formal class of engineering design criteria process, which is actually very different than like the typical scientific method we see or what we were taught in high school and middle school with like hypothesis. Like I would say those are more structured, but with the engineering process, it is so open-ended in the fact that, when one solution doesn't work, you still have to find a way to deliver the solution to the client. So you have to look at other prototypes. There's a lot of iterations, and most importantly, collaboration. So that's the thing that jumped out to me when I was doing this EGR 101 class, like how to not only collaborate with others, but improve my hardware set of skills like soldering, 3D printing, which I was already familiar with prior, RGB circuits, and laser cutting. And another important thing to note that as an engineer, you'll be dealing with very fun but academically rigorous prerequisite courses like linear algebra or matrices and vectors, often to a higher extent. So I remember one inside joke with all of our engineering students is we really loved our matrices and vectors class, but we really had to like study to be on top of it. And, at first it doesn't seem like it's relevant at all. But, when I've been looking at my course syllabus for the next 4 years at Duke, I've realized, especially in the hardware aspects of BME and MECE, you kind of need these fundamental from multivariable calc or physics, mechanics, and electricity and magnetism especially. And so it's also been a very interesting experience combining my engineering education, which is kind of a bit technical, with the other engineering students I collaborate with my pre-medical studies, through like just taking general chemistry right now, and the next semester I'll be taking orgo. So I'm really just excited to see how these two will mesh together as my college progresses.
Right. So this is sort of a heavier question, but where do you see yourself in the next 10 years?
It's so funny because I've just seen this question a lot, and it's like often at times, especially in high school, it's like, oh, I'll do this. Like at first, I wanted to be like an MD PhD because I really love research a lot. But as I've started, engineering, especially where I'm more very involved with industry afterwards, I think my current goal right now is to take advantage of Duke's 4+1 BS MSc program, which basically allows me to get a master's in any field of engineering right after my undergraduate studies at Duke within 1 year, and then hope to move on to medical school. The reason why I really chose to major in biomedical engineering was that in the next 10 years, I really want to be like a doctor, that not only helps treat and care for patients, but also can act as a bridge between emerging or innovative medical technologies, the biotechnology realm, with how doctors can use this technology and become more, I would say, knowledgeable, but also sophisticated, more knowledgeable about how to use this sophisticated technology in the future of healthcare, and basically hopefully be involved as like a chief medical officer within some sort of biomedical engineering company or biotechnology company. As I really think that, especially with the way technology is progressing, we'll see a lot of personalized healthcare being this sort of focus through manufacturing of various devices or targeted BME solutions.
Wow, that sounds amazing. All right, so that is all we have for today. Perisa, thank you so much for your time and for sharing all of your, wonderful experiences with your science projects and, your future plans. We really, really appreciate having you on our podcast.
Thank you so much, Mrunal. I had a wonderful time sharing my experiences, and both with science fairs and at Duke. And thank you so much for this opportunity.
Yep. All right, so for our next podcast, we will have David Kang, co-founder of STEMinate and current Harvard student, speak about his STEM experiences. See y'all next time.